All right, moving on to bulk transport. Sometimes we need to bring in big stuff. This is an active process. It is going to require energy because we are going to have to synthesize vesicles. Exocytosis is when products are released. Endocytosis is when nutrients and substances are brought in. And like I said before, this does require transport vesicles. So looking at exocytosis, these are going to be in tissues that release cell products like glands. Think of salivary glands that are releasing enzymes, nerve cells that release neurotransmitters, plant cells secreting cell wall material, and a host of whole other cells. And we're also going to use this process to change the membrane proteins on the cellular membrane. We can put those into the vesicle membrane at the Golgi, and when the vesicle fuses with the cell membrane, we can change that composition. We can add up channel proteins, we can change up the glycoproteins, we can even change the enzymes on the surface. Any of those proteins embedded in the membrane can be changed by putting them into the vesicle so that it fuses. So when the vesicle fuses, the substances that it carries is going to be released into the extracellular fluid and taken wherever it needs to go. They will be usually tagged with some molecular tag and not just randomly left to go wherever they please. Endocytosis is going to bring substances in. Phagocytosis brings large solid particles in. This is usually called cell eating. Macrophages, your white blood cells that engulf bacteria. Amoebas, single cellular protists that engulf other small organisms to bring them in. And when phagocytosis bring them in, that food vacuole is going to fuse with a lysosome that has the hydrolytic enzymes, and it's going to break down what cellular material it can to use those in building other cellular products. Sometimes the cell needs to bring in fluids or dissolved substances, and this is called pinocytosis. And the cells are going to form vesicles at the surface of the membrane and pinch in. They're going to be coated with proteins on the underside to trigger the vesicle formation. And the last kind of endocytosis is called receptor-mediated endocytosis. This is when you have something that the cell needs in such a small amount that it has to put up receptor proteins to capture it. It's kind of like fishing for the molecule. So it's used to bring in very targeted substances. And when the receptors have captured their molecule, it's going to tell the proteins on the other side to form the vesicle. And it's called a coated pit because of the red proteins that coat it and the receptors on the other side. So it uses coated proteins to trigger the vesicle formation. And an example of this are LDL receptors. Animal cells need cholesterols to maintain membrane structure. Well, LDL receptors are what brings those cholesterols into our cells. And this brings us to the end of our presentation on cell membrane structure and function. We're going to come back to a lot of these processes throughout the other parts of this course and future courses. All of this ties in together in how organisms work and function.
What you should have learned in this segment is the components and function of a cellular membrane, the function of those components like diffusion, facilitated diffusion, and active transport, how osmosis works, how we move water across the membrane, what happens to a cell in a hypotonic environment and a hypertonic environment. This brings us to the end of our biochemistry unit. You have a quiz over 1.1 all the way through 1.4 next week. So be sure you go back and you review all that material. Good luck to you.